or by replacing the intervertebral disc with artificial disc. So, as I showed later, previously, this is actually the current clinical treatment, and what we are trying to do is to isolate from the adipose tissue and the bone marrow the mesenchymal stem cells into culture, and then engineer them with BMP2s or BMP6, and then re-inject them at the site of that we are interested to induce spinal fusion, and we hoped that the bone formation induced by these cells at the paraspinal region can induce the fusion of the two vertebrae. And as you can see here in a mouse model, we could see that following the injection of MSCs expressing BMP6, we could achieve the fusion of several vertebrae. Here is in orange, you can see the bone formation that was induced by the cell injection, and here you can see the cross sections showing the fusion of the bone that was formed with the native bone of the vertebra. So this was our first application for spinal disorders, and we later went on and tried to repair the intervertebral disc tissues itself. If we look at the intervertebral disc, it's actually composed of two tissues. The first is the surrounding tissue that is called the annulus fibrosus. This is a tendon-like fibrotic tissue that quite resembles the tendons found in the skeletal system, and the inside of this tissue is actually called the nucleus pulposus, and this is a kind of cartilaginous tissue that this part of the intervertebral disc actually functions as the shock absorber. So our approach was that if we could intervene using MSCs to repair one of these tissues after injury. So here you can see using MRI that the degeneration of the disc first starts with damage to the annulus fibrosus tissue, and then this is followed by either the herniation or the depletion of the nucleus pulposus tissue that later caused the direct contact of the two vertebrae. And in this study, we first we utilized our findings using the SMAD8 signaling molecule. This is a molecule that we found to have function in the BMP signaling pathway, and we found that if we express this molecule in combination with the BMP2, we could find that the cells differentiation is actually going toward the tendon lineage. We can see here that in vitro these cells that express the BMP2 and SMAD8 express many markers that are related to tendon formation, and in vivo we could see using MRI images performed at the Achilles tendon injury model, we could see that these cells can actually and functionally repair this defect. Here is just as we followed the cells engraftment using luciferase imaging. This is also another type of MRI images showing that, as you see here in the white color, the repair of the Achilles tendon injury that quite resembles actually the intact tendon. This is this is actually very new data that we are speculating regarding the aging of the intervertebral disc tissue, and we first found in a collaboration with Jefferson University, we found that the nucleus pulposus tissue contains stem cells that much resembles the phenotype of mesenchymal stem cells, and you can see here that these cells can be isolated from degenerated human discs and also from healthy rat discs. And here we hypothesize that if the degeneration of the intervertebral disc is related to these cells, it might be that this is due to the depletion of the number of these cells. It might be because of the lower proliferation or apoptosis of these cells. This is very fresh data. Just confirming these results, we now could isolate the NP cells from pig's intervertebral disc and differentiate them in vitro to 3 tissues. Just to summarize this talk, we can say that MECs can be applied to induce spinal fusion, and we can say that MECs are potential candidates for the regeneration of intervertebral disc tissues, and tissue-resident MECs could be a potential target for therapy in order to prevent the disc degeneration.
Thank you.